Assalamualaikum. Welcome to Nasra Public School online teaching system. Now this is the class 9th and subject biology week 2 and lecture 1. We are going to start our chapter number 2, that is structural organization of life. Here are objectives that we are going to study. Number 1, microscope. Number 2, concept of light microscope and electron microscope. And third one is the magnification and resolution. So this is microscope and we are going to start definition of microscope. So microscope is an instrument that is used for the study of microorganisms and cells. It is an important tool which magnifies many times to their original size, which cannot be seen by naked eye. So here are types of the microscope. Basically there are two types of microscope. Number 1 is light microscope and number 2 is electron microscope. This is simply the diagram of microscope. Number 1, that is light microscope. Here are the definition we are going to study. Such microscope in which visible light is used as a source of illumination and glass convex lenses are used for magnification is called light microscope. Here is the word, that is illumination. Illumination means light. And there are types of light microscope. Number 1 is light simple microscope and number 2 is light compound microscope. So that is number 1, light simple microscope. So question arises, what is meant by light simple microscope? Here is the definition. You can see such microscope in which only one lens is used for magnification is called light simple microscope, which was first invented by Galileo in 1610. And now second one is light compound microscope. Light compound microscope was first invented by Leeuwenhoek in 1660. A microscope in which more than one lenses are used for magnification is called light compound microscope. It can magnify the object up to 1000 times, but its resolving power is very limited, only 0.2 micrometer. Here you can see the symbol U, small um. You can read it micrometer. This is the symbol of micrometer. Okay. Now obviously you are familiar about what is the, you can say what is the difference between light simple microscope and light compound microscope. In light simple microscope only one lens is used for the magnification, and in light compound microscope one or more than one lenses are used for the magnification. That is called light compound microscope. So we are going to start magnification. It is the power of a microscope that how much it can increase the size of an object. Next one is resolution. The minimum distance of object is called resolution. So this is the difference between resolution and magnification. You can see what are the difference between both of them. Number 1, resolution, and number 2, magnification. Number 1, resolution. Resolution is the capacity to separate adjacent objects. At the other side, or you can say on the other side, magnification. Magnification is this is the spelling mistake, means of increasing size of an object. Second point is the resolution is maintained up to certain magnification. On the other hand, by increasing magnification, resolution is disturbed. Number third point of resolution is it improves as the wavelength of illumination becomes shorter. On the other hand, magnification improves with the focal length of lens. Now, number 2 is the electron microscope. It is the most advanced form of microscope. It was invented by scientists in 1935 to increase the resolving power of microscope. Electromagnetic condensers are used as lenses instead of glass lenses. Screen or photographic plates are used to see and focus an image. The electron beam increases its resolving power 1000 times or more than that of a light compound microscope. It can achieve a resolution of about 0.2 mm. Electron microscope reveals all such organelles of cells which are not seen with light microscope. This is the picture of electron microscope, and you can see each parts of electron microscope. For example, electron gun you can see, vacuum chamber you can see, anode, condenser lens, electron beam, objective lens you can see, backscatter detector, secondary detector, sample chamber, X-ray detector, sample. You can see all the parts of electron microscope. Now we come to the difference between light and electron microscope. Light microscope, it is small in size and can be carried easily. On the other hand, electron microscope, it is large in size and operate in a special room. In light microscope, a ray of light is used for illumination. Illumination means light. On the other hand, electron microscope, a beam of electrons is used for the illumination. Here is a word beam. Beam means ray. Okay. Now third point is in light microscope, glass lenses are used, but on the other hand, electromagnetic condensers are used. In light microscope, it is easy to operate. On the other hand, it is complicated instrument. In light microscope, it is not affected by electromagnetic field. On the other hand, in electron microscope, it is affected by electromagnetic field. Now let's move to the video. It will definitely explain you more about microscope and types of microscope.
While the telescope has made it possible for us to view the cosmos, the microscope has allowed us to see the smallest components of all living things. Magnifying glasses are mentioned in the writings of Roman philosophers during the 1st century AD. But it wasn't until the 17th century that Galileo, the father of modern physics and astronomy, formulated the principle of lenses and focusing. Robert Hooke built the first usable British compound microscope in 1665. It was a light microscope with two converging lens systems, the objective and the eyepiece. Hooke observed that tissues were constructed of many tiny individual building blocks. In 1665, he had discovered and named the cell. Hooke was actually viewing cork, dead plant cells. The first person to witness a live cell under a microscope was Anton van Leeuwenhoek. Around 1670, Van Leeuwenhoek developed a method of grinding very thin glass lenses, just a millimeter in diameter, and yet they could magnify by several hundred times. These single lens microscopes were far superior to the early compound instruments. Mounted in a brass plate, These lenses could use transmitted light to image objects in a drop of water on the end of a metal pin. A screw was used to move the pin and focus the specimen. Van Leeuwenhoek was the first person to see and describe bacteria and yeast cells, sperm and blood cells, and witness the circulation of blood through capillaries. Light microscopes are still used today, but in 1931, German scientists Max Noll and Ernst Ruska invented the electron microscope. Instead of light, this microscope employs a focused beam of electrons to image the specimen, and is capable of far greater magnification and resolution. The electron microscope can magnify up to 1 million times, making it possible to view objects as small as the diameter of an atom. These powerful microscopes have one major drawback. Living specimens are destroyed by the high radiation of the electron beam. So scientists must continue to rely on traditional light microscopes to examine living cells.
So this is the summary of your chapter, or you can say lecture. Microscope is an instrument used for the study of cells. We have learned. Okay. Two types of microscope: light microscope, electron microscope. Cells are observed under light and electron microscope. Two types of light microscope: light simple microscope, light compound microscope. Difference between magnification and resolution. And last one is difference between light and electron microscope. These are all topics that you have learned in the lecture. I hope you like this video. Thank you so much.